Hi everyone. Very all tricky Tuesday. So yesterday we have seen about the DNA structure, isn't it? And I posed a question also to think, and you come with the answer. I said, what is that question? Why DNA is an acid, isn't it? So why it is an acid, children? It is an acid because it has a phosphoric acid in it. That's why this phosphoric acid, no, it is in the form of phosphate in DNA. So this phosphate only will incorporate or will import clear. That's why due to the presence of this phosphoric acid in the DNA makes the DNA acidic in nature, and this DNA can donate the proton to others. So that's why space but we're going to see today the double helical structure model proposed by Watson and Crick. Okay, so this Watson and Crick, no, they uh, discovered the DNA double helical model. So, what are all the points they discovered in their discovery? So, we are going to see that in an elaborate form in this session. Okay, so now if you are new to the channel, please subscribe and then share my videos to your friends also. I'm taking the class 10 genetic topic. In that, a small chunk I have chosen, that is DNA structure and its model. So yesterday we have seen about the DNA structure basic model. What is the structure? So that video also I will give you the link along with this video. And then today we are going to see about the DNA model proposed by Watson and Crick. There's a double helical structure. Okay, so now let me explain one by one. So it will be very useful for you to prepare for your exam. So if you look at this DNA structure, no, see, this is the double helical structure. So it will be many in number, means what? Millions of nucleotides will be present in the living cells. So that is why this DNA is called the double helical structure. So if you look at the structure, no, I told you if it is a only one strand, one double strand is there, means it is known as the nucleotide. So we have seen what about the nucleotide in the previous session. The nucleotide means what? The nucleotide will have the sugar, then it will have the nitrogenous bases, and along with that it will have the phosphate group. That is the nucleotide. So this is more in number, means that is known as the polynucleotide. That's what Watson and Crick know. They said the first and foremost discovery they did is this DNA double helix is a polynucleotide. Is that clear? So DNA molecule consists of two polynucleotides. So one will be going in one direction, the another one be in the opposite direction. So that's known as the polynucleotide. And the second thing is they said it is a double helix. No, double helix means two helical structures. Helix. So if you look at this picture, no, this is the helix. Okay, so this is one turn. Okay, and then this is another turn. This is the double helical structure of DNA. Is that clear? And the third point, what they said, Watson and Crick in their discovery, no, they said that the DNA molecule, no, they have a nitrogenous base in the center. So what is the nitrogenous base? Yesterday we have seen, no, they are the purines and pyrimidines. So what are the purines? Adenine and then the guanine. Then the cytosine and the thymine are the that is pyrimidines. So purines are adenine and guanine. Pyrimidines are cytosine and the thymine. So these bases they will form a bond between each other. What they bond, how they bond with each other, with the hydrogen bond. So these green color bonds they have given, no, that is the hydrogen bonds between the nitrogenous bases. So Watson and Crick they said about the nitrogenous bases in the center, and they will be linked to the sugars in the backbone of the double helical structure, and along with that phosphate also linked. So that is the sugar and phosphate along with the nitrogenous base, it will form a polynucleotide. So let me explain in detail about this, children. Now come, we have seen the structure yesterday also. Is that clear? Yesterday also we have seen no this structure. So what is this? This structure is mainly the double helical structure I have given in a what to say structural form. That is chemical formula I have given here. So this pentose ring is the sugar. This pentose ring is known as the sugar. What is the sugar? It is the deoxyribose sugar. Is that clear? And this deoxyribose sugar will be bonded with the nitrogenous bases in the center of the DNA. In the center of the DNA. So this whole structure is the DNA. I have shown a small fragment of the DNA only here. So this nitrogenous bases will be in the center, and they will be bonded with the sugar, and this sugar in turn will be bonded with the phosphate group. So that's why DNA is called a nucleic acid. Yesterday we have seen, no. So it is an acid because this acid, phosphoric acid, only gives the acidic nature to the DNA molecule, so that they can donate a proton, they can donate an hydrogen atom. Proton means hydrogen atom. Is it clear? So what's the third point of the Watson and Crick? The Watson and Crick proposed this sugar will linked with the nitrogenous bases in the middle, that is in the center, and this sugar in turn will be bonded with the phosphate group. Is that clear? That is the third point proposed by Watson and Crick. And not only that, these two chains, no, these two strands, so they will run opposite to each other. If one will be in one direction, the another one be in the opposite direction. So this is known as the anti-parallel. The two strands are anti-parallel to each other. Is that clear? And then if you see this, always the pyrimidines and the purines, they will be bonding with each other. What is that? The cytosine will be bonding with the guanine, and the adenine will be bonding with the thymine. So adenine is the purine, thymine is the pyrimidine. Similarly, cytosine is the pyrimidine, guanine is the purine. So they will be bonded with each other in with a hydrogen bond. If you see, there is the three lines between this C and G, no, between the cytosine and guanine. How many lines are there? There are three lines. These three lines are known as the hydrogen bonds between the nitrogenous bases. And then if you look at this adenine and guanine, there will be two hydrogen bonds between the adenine and guanine. So this is double bond. Okay, double hydrogen bond between the adenine and guanine, thymine, and then cytosine and guanine will be a triple bond. And then Watson and Crick, what they are coming to say with this hydrogen bonds, these hydrogen bonds only will make the DNA molecule more stable. Okay, without these hydrogen bonds between the nitrogenous bases in the center of the DNA, this DNA molecule will not be stable. What do you mean by stable? What is stable? So today homework, I'm going to give you the stable. What is the stable meaning? So find out and come when you come to the next class, so that we will see what is the stable answer. So that Watson and Crick, what they said, due to the presence of this hydrogen bond between the cytosine and the guanine, and then adenine and the thymine, they are they are linked with each other with the hydrogen bond. That hydrogen bond makes the DNA more stable. Clear? And then always adenine will be pairing with thymine, and then cytosine will be pairing with the guanine. So if you see, no, this base pairing adenine with the thymine and guanine with cytosine is known as the complementary base pairing. Understood? So in the DNA molecule, there will be always the complementary base pairing. Is that clear? So this is another point proposed by the Watson and Crick. And not only this, they have proposed one more point. What is that one more point? This there will be the distance, no, the double helix. Each turn. This is one turn, and this is another turn. So the distance between this turn is the 3.4 nm. What is that? It is the 3.4 nanometer. Understood? And then the distance from the center to the corner, it is known as the 1 nm distance. Understood? And then the distance between this helix, this one turn of helix, no, that is known as the 0.34 nm. Is that clear? So now, and also Watson and Crick, what they said during their discovery, the complete turn, no, one complete turn of a DNA molecule will have 10 base pairs. So in this one turn, no, there will be 10 base pairs. Base pairs means this purine and pyrimidine base pairs. Understood? That is the base pairing means nitrogenous base pairing. So one complete turn of a DNA molecule helical structure will have 10 base pairs in it. Is that clear? That's another one proposed by James Watson and Francis Crick. Hope I'm telling correctly and clearly to you. If you have any doubts, you can comment me after this video also in the comment section. This video will be in the channel, okay? Or I will share you the link also, okay? So this is about the next point in the Watson and Crick model, and the last and the most important point what Watson and Crick said, no, it's nothing but the phosphodiester linkage. So the sugar and the phosphate, they are linked with each other, no, that's about the phosphodiester bond. Clear? So I told you very clearly that DNA molecule is a simple double helical structure, and it is a hereditary information. So from that only the what to say, the genetic information is transferred from DNA to the next generation. So we are studying about the structure of DNA only. So in the DNA molecule has a polynucleotide, and it has two strands. Each strands are running in opposite direction, and then in the center of the DNA you have nitrogenous bases. These nitrogenous bases are bonded with a hydrogen bond. This hydrogen bond only makes the DNA more stable. And not only that, this nitrogenous bases are attached to the or linked to the sugar, and this sugar is in turn is linked to the phosphate group. This phosphate group only gives the DNA a acidic nature, and in turn this sugar is also linked to the another sugar molecule through this phosphodiester bond only. So it is a continuous linkage. So when one strand runs in one direction, the opposite strand will be in the another direction. So this is known as the anti-parallel strands. Okay, they will be in anti-parallel. For example, this is 5 dash to 3 dash means this will be 3 dash to 5 dash. Understood? So this is anti-parallel with each other, and the bases are complementary base pairing. That is always cytosine will be pairing with guanine, and then adenine will be pairing with the thymine. So they are known as the complementary base pairing. Is that clear? Now we will come to the this complete recap of this session. Before that, I'm going to tell about the Chargaff rule. So, what is Chargaff rule? So, this is nothing but a scientist known as the Erwin Chargaff. He states that always there will be the equal proportion of the purines and pyrimidines. That is, if always adenine will be equal to thymine, and then guanine will be always equal to cytosine. There will be equal proportion of purines and pyrimidines. So this rule is known as the Irwin-Chargaff rule. Okay, so since it was found, or it was discovered by the Erwin Chargaff scientist, this rule is known as the Chargaff rule. So the Chargaff rule always states that there will be equal proportions of purines and pyrimidines. Is that clear? So adenine will be always equal to thymine, and guanine will always be equal to cytosine. So adenine and guanine are purines, thymine and cytosine are pyrimidines. Okay, so now we will see the Watson and Crick model in detail. That is, let me show a flow chart of the Watson and Crick model so that you will understand better. So, okay. So now look at this DNA Watson, that is Watson and Crick model of DNA double helix. So what is the first point we have seen in this session? DNA is a polynucleotide chain. It has two polynucleotide chain. What is polynucleotide? Many number of millions of polynucleotides are present. So that is known as the two polynucleotide chains will be there. And then the second point, what we have seen, double helix structure. There will be double helical structure with the two strands. Is that clear? There will be a double helical structure with the two strands, and then it will, the two strands, no, they will run in anti-parallel direction. Just now I told you, if one strand is going in one direction, the another strand will be in the opposite direction. That is anti-parallel. Understood? So they will not be going in this together in the same direction. One is going in one direction means the another one will be coming to the opposite direction. See, like this. So if one is running in one direction, that is going upward, the another strand is coming will be in the opposite direction. This is known as the anti-parallel. Understood? So the two strands are anti-parallel, running in anti-parallel direction. And then not only that, what we have seen in this, nitrogenous bases, no, they are present in the center of the helical structure. How they are linked, to which substance they are linked? They are linked to the sugar phosphate linkage. So this sugar phosphate only, they will form the backbone of the DNA. Is that clear? And then we have seen about the DNA pairing between nitrogenous bases is very specific. So I told you that adenine will be pairing with thymine, and the guanine will always pairing with the cytosine. That is very specific. Base pairing in between the nitrogenous bases is very specific. They will not change. The adenine will not pair with the that is guanine, and the thymine will not pair with the cytosine. So they are very specific. A will be pairing with the T only, and C will be pairing with the G only. So that base pairing is known as the complementary base pairing. Is that clear? And then what we have seen in this, the DNA, the hydrogen bonds between the nitrogenous bases make stable DNA. I told you, no, there's a double bond between the adenine and the thymine, and the cytosine and the guanine will have the triple bond. So this hydrogen bond between the DNA only makes the DNA more stable. So I have given you one task that we have to come out with the answer for stable. Why it is called stable? Okay, please go and check and come. And then we have seen one more point, that each turn of the double helical structure which was proposed by the Watson and Crick is 34 Armstrong, 3.4 nm. Understood? So, and a complete turn will have the 10 base pairs. What is the base pairs? Nitrogenous base pairs only: adenine, thymine, cytosine, and guanine. So there will be a complete base pairs will have 10 base pairs in it. A complete turn, sorry, a complete turn will have a 10 base pairs between them. Understood? And then last thing is, we have seen about the nucleotides are joined by phosphodiester bond. Is that clear? So the nucleotides, how they are bonded with each other like a chain, like a strands, they are bonded with a phosphodiester bond between them. So there are two types of bonds in this DNA molecule. What is the one? The one is the nitrogen, this hydrogen bond between the nitrogenous bases. Another one is the phosphodiester bond. Is that clear? And then what type of sugar the DNA molecule has? It has the pentose sugar. Understood? And this pentose sugar will be connected with the phosphate group, and this phosphate group, in turn, they are linked with the strands by a phosphodiester bond. Is that clear? So this is about the complete what is the picture of the DNA Watson and Crick model. So I will post this in the community also. You can take the picture so that it will be useful for you. Okay, children. I hope all the concepts are very clear. If you have any doubt, please come out with your comments so that it will be helpful for me. And you can also give your feedback, valuable feedback about my teaching so that I can improve myself in the upcoming session. Yes. So today the session went very well, and then tomorrow let me explain the DNA replication process. So we will see what is replication. Please go and refer what is replication, come back so that you can also comment me during my class. And this class mainly for the class 10 students, or also those who are preparing for the there's a biology group, biology students in the +1 +2. For them also it will be very useful, this genetic topic, because this is a general topic I am taking in the genetic, that is the DNA structure, basic structure, and what are the parts of the DNA, and then who proposed the DNA, and then how was the structure of DNA was discovered by the great people. And today we have seen about the Van Crick model of DNA. Is that clear? So you can share your comments, and then you can share this video among your friends also, so that it will be useful for you in the upcoming session. Okay, so see you tomorrow. Thank you. Have a nice day, and then come up with the answer for the table which I have given today. Thank you.